we're going to get started. Sorry, I just skipped this thing. Thank you everyone for joining Grand Rounds this morning. I'm going to start with our land acknowledgement. Ottawa is built on unceded Algonquin Anishinaabe territory. The peoples of the Algonquin Anishinaabe Nation have lived on this territory for millennia, and we honor them and this land. Their culture and presence have nurtured and continue to nurture this land. CHEO also honors all First Nations, Inuit, and Métis peoples and their valuable past, present, and future contributions to this land. The presentations today are from the CHEO Precision Child and Youth Mental Health team. Dr. Kathy Pager, the Medical Director of the Precision Child Mental Health Initiative, will be introducing the panel of speakers.
Thanks very, very much. And let me just minimize this so that I can see all the slides. Okay, so thank you all for coming this morning. We are going to talk about Precision Child and Youth Mental Health Initiative. Are we there yet? So spoiler alert, the answer is absolutely not. The only reason that I put it up there is because, and named it thusly, is because when we tell people about it, they're so excited. They said, well, is it going to happen like tomorrow? Like next year? Is my kid going to be able to get this? And we have to say, this is going to take a little while, but we have made progress. So today, 3 speakers who are representing 3 of the grants that were given out by Precision Child and Youth Mental Health, 3 of 7, will be presenting brief overviews of their work, and then I'll moderate a discussion and question answer period. There are, you heard from Mark Norris last month, I believe, in a Pediatrics Grand Rounds about some of the Precision Child and Youth Mental Health, and we refer to it as PCYMH, so that's what you'll hear from now on. Some of the PCYMH funded work that they're doing in eating disorders. And then in January, one of our other researchers, Marc Tremblay, will be giving grand rounds on the work that he's done. And that and the remaining 2 people who are still a little further, a little not quite as far along, will be giving overviews of what they've learned probably sometime in March. Okay, so what's the problem we're trying to solve? I don't know if you folks know this, but child and youth mental health treatments only work in about 50% of the cases if you can imagine a big meta-analysis of every single kind of disorder. What's the consequence of that? First of all, prolonged suffering for patients and families. Second is disruption of normal development. And the third is adult dysfunction in all domains, from untreated mental health disorders. There are system consequences of the problem too. Untreated mental health problems often become emergencies, as we are very well aware. Patients with untreated mental health problems can't really get out of care because they don't get better. And they remain in the system, which winds up being that we don't have enough providers to take care of everybody, and the whole system winds up in gridlock. So what is precision child and youth mental health anyway? It's more precise mental health care for each patient, accomplished by collecting data and integrating it on biological aspects, for example, genetic, metabolic, brain function, lifestyle, and environmental characteristics. And you add those to what we usually deliver, our best attempt at personalized care, which is symptoms, and it doesn't wind up being very personalized. That's the problem. So you add all these, and then we can match these characteristics and profiles of them to treatments. So some examples are, and these are real examples, using psychopharmacogenomic informed antidepressant selection for Kayla's treatment-resistant depression, or ameliorating Jean-Paul's long-standing ASD symptoms that were treatment resistant with biomarker study following by genetic testing that revealed a genetically based cerebral folate deficiency, which could be successfully treated by leucovorin. Each of these patients until that point in time had received what we do when I say standard sequential treatment regimens. That makes it sound that it's really very well thought out and organized, but it's just that we kind of try everything in a certain order, but we kind of try everything. And we do it for everybody till they get better. And this one size fits all just does not work. So let me tell you just a little bit about the CHEO, Precision Child and Youth Mental Health Care Initiative. It is an initiative we share with Sick Kids. Together, we got a $10 million donation that began in April of 2021. It was split evenly between the two sites. We do some work together, and we built at our each site. So some of the things that you see here, this is our annual report from April of 2023. Sorry, it didn't begin in 2021. It began in 2022. We started negotiating in 2021. So we have built, and I know this phrase is overused, world-class, but I'm telling you, it really does deserve to be used here. We've built a world-class high-performance computing platform for the CHEO-RI, for the infrastructure to do precision child and youth mental health. And one of the other things you see there on the bottom left is that we gave out 7 grants, $1.3 million worth of grant funding. And that's some of what you're seeing the results from today. And the other thing is to just point out that we are not only doing what you'd call straight research, but we have right from the beginning worked at the front line with our clinicians and administrators to start changing and do clinical transformation to set us up for precision child and youth mental health. So without further ado, I am now going to just let you know, we are doing a 2024 Precision Child and Youth Mental Health Conference, June 10th and 11th at the Shaw Center. It's an international conference. So, okay, wait a minute. I am very sorry. Hang on. I had carefully set everything up and I did it incorrectly. Very sorry. Oh my gosh.
Kathy, did you want me to share my screen?
Well, wait a minute. I have it now. I just had put below the wrong one. I'm very sorry. I usually do not mess up this way. Okay, now let's just go through. Sorry to make you look at this again. I just want to make sure. Oh, there it goes. There is Addo's. So there you go. Sorry about that. I hadn't realized I did that. All right, I'm turning it over to Christina and Addo. And you just tell me when you want me. Do you want me to hit next now?
Oh yeah, my name's Christina Canton. I'm a research coordinator with the mental health team at the Research Institute. So I'm presenting on behalf of Addo and our team the chart audit of pharmacogenomic testing in a pediatric psychiatry. What are the implications for psychotropic medication selection? So next slide, please. And we have 3 learning objectives. We hope by the end of the presentation, you'll understand why pharmacogenomic testing was offered to adolescents seeking care for psychiatric disorders at CHEO, specify the proportion of adolescents who were at risk for poor therapeutic response or adverse outcomes, and identify the potential opportunities to incorporate precision medicine as a strategy to inform clinical treatment options. So next slide, please. We have a large team that includes the CHEO psychiatry, so 4 psychiatrists and a pharmacist, other CHEO units, including the clinical research unit and genetics, and some external partners at the University of Ottawa, Institute for Mental Health Research, Dynacare Lab, as well as 2 patient partners. Next slide, please. In Canada, nearly 1.2 million children and youth experience mental health issues. And early intervention is important given that untreated depression in children and adolescents is associated with an increased risk of substance abuse, reduced academic and social functioning, and suicidal behaviors. And in 2019, in Canada, among youth 15 to 19 years of age, 140 males and 81 females died by suicide. And so tragically, suicide accounts for 25% of all deaths among 15 to 24-year-olds in Canada. The treatment for psychiatric illness has traditionally utilized a wait and see or a trial and error approach. And this can result in delayed effective treatment, suboptimal clinical response, increased side effects, and medication failure. As Kathy mentioned, among children who start medications, up to half will not respond to the medication, and up to 25% will experience an adverse drug reaction. So next slide, please. One potential reason for this is related to genetic differences in how the medication is transformed in the body. So on this slide is a picture of chromosome 17 and the SLC6A4 gene with the variation in the promoter region. So at the very left end of the gene. This impacts serotonin transporter, which influences the movement of serotonin in the synapses. So pharmacogenomic testing assesses gene level variation that affects how medication works on the body of a particular person. So that's called pharmacodynamic genes. And then how the body breaks down or metabolizes medication, and that's called pharmacokinetic genes. So integrating this testing might speed up treatment time by adopting a personalized medicine approach by selecting the appropriate medication, minimizing side effects, and preventing medication discontinuation. So genetic testing is a promising strategy to help healthcare providers choose the best medications for children and adolescents with major depressive disorders. However, more research is required, as Kathy mentioned. Next slide, please. We have a lot of data about pharmacogenomics, and this slide was recently shared at the World Congress for Psychiatric Genetics in Montreal, and I'm using it with permission from Dr. Chad Boosman from the University of Calgary. So we have data on 7 pharmacogenomic genes as it relates to 30 medications, and you can see antidepressants in the blue oval and the associated genes that are involved in metabolization. So primarily the CYP2D6 and CYP2C19. Next slide, please. We conducted a retrospective chart audit study to determine the frequency of 6 serotonin transporter genotypes in a sample of participants at CHEO with depression. And we compared this genotype frequencies that's seen in the general population. We also wanted to determine the likelihood of increased risk for adverse events or poor response for 3 groups of antidepressants and 11 commonly prescribed antidepressant meds for different gene variants. So next slide. A total of 119 eligible participants were identified by CHEO psychiatrists. And after 4 duplicates were removed, the total sample consisted of 115 children and adolescents. So participants ranged from 10 to 18 years of age and had a mean age of 15 years. The clinical diagnoses were primarily anxiety and depression, both at 83%. Next slide, please. We found in terms of the frequency of serotonin transporter polymorphisms that 27% of participants had the SS genotype. And this is associated with poor medication response and increased adverse outcomes. So this is the box in red. Studies have repeatedly shown that the short variant, the S, is associated with a reduction in the expression of the serotonin transporter, resulting in reduced reuptake of synaptic serotonin. And compared to the LALA in the green box, these individuals are having the best or most favorable medication response and fewer side effects. So to answer the second part of our first research objective, the statisticians on our team conducted chi-square tests of variant prevalence between our study results and those of populations in 4 other published studies. And the results indicated that there was no difference found between the prevalence of variants in our study compared to the other populations. So next slide. Answer our second research objective. We analyzed the results of the CYP450 drug metabolism to determine the risk for adverse events. And you can see that for 2 of the CYP chromosomes, so 2C19 and 2D6, that some of the participants were noted to be poor metabolizers. So that's in the red font. Most participants were noted to be normal metabolizers or intermediate metabolizers. So next slide, please. There were some limitations to our study, namely a small sample size of only 115. And although ancestry information was on the requisition, we did not have ethical approval to include this in our analysis. However, after consulting with experts in pharmacogenomics, we have some potential additional testing and analysis for which we will seek REB approval. And we hope that this additional analysis will provide insight into our study findings. Another limitation is that the study participants paid for the genetic test. So the sample was biased towards families who could afford this test at a cost of about $500. So next slide, please. So we have identified an opportunity to use precision medicine to potentially improve care for adolescents who are at the greatest risk for suboptimal medication response and adverse outcomes. So those are the individuals with the SS serotonin transporter variant. We have already formed another strong research team with diverse clinical, methodological, and research expertise. We have prepared a protocol for a pilot randomized control trial. And a large part of the budget is allocated to paying for the genetic test to ensure equitable opportunity to participate in this research. For the trial, we are proposing to compare an SSRI medication, so that's a selective serotonin reuptake inhibitor, which is currently recommended as the first-line treatment for depression in adolescents. So we want to compare the SSRI with an SNRI, a serotonin norepinephrine reuptake inhibitor. And the hypothesis is that using a medication with a different pathway that doesn't involve the serotonin transporter might result in a favorable medication response. So at the moment, we're consulting with patient and family advisors and other stakeholders to obtain feedback on the proposal to ensure that the information is clearly presented so adolescents can make an informed choice about participating in the study or not. And we're obtaining feedback on our recruitment strategies. Next slide, please. So that
Thank
is
you
the work
very much.
that we have been doing so far, and we'll answer questions at the end.
So we're going to hold questions and comments to the very end. Thank you. So now we're going to go on to hear Kevin Dick. Yes, we are going to go on to Kevin Dick. Perfect. There you go, Kevin.
Excellent. Thank you so much for this opportunity. So yeah, hi everyone. My name is Kevin Dick. This work, we call it the ELEGANT study. So it stands for Precision Mental Health Using Artificial Intelligence for Early Intervention. Much of this work was done while I was in my postdoctoral position through CHEO. I've since moved on to become an AI data scientist at Bourne, Ontario. But I still hold cross affiliations with CHEO and OHRI, and I'm excited to present this work to you all. So next slide, please. So our work really focused around autism spectrum disorder, otherwise known as ASD. I'm sure most of you are familiar with this. We know that it is a complex neurodevelopmental condition, but what's exciting is with early detection, we can allow for early intervention. And thus, the whole question of how we can accurately identify individuals early on, the hope is that through early diagnosis, we can then facilitate timely access to resources to support that individual, their development, their growth, and the families. However, most of the time, diagnoses might potentially be missed or delayed since to date, ASD detection really operates by recognizing symptomology. So the real kind of impetus behind our work, the question we wanted to really address was whether we could leverage kind of these large scale data sets, so registry and health administrative data sets to potentially develop a screen for young children. We really want to hopefully discover a way or identify a way to find those individuals that might be at increased risk for developing ASD downstream. And the idea was, can we use machine learning, or it's kind of subfield deep learning? I know there's a lot of AI hype going on right now, but the idea is, can we use these kind of new state-of-the-art AI methods to that end? So next slide, please. So if you're not familiar with kind of different types of machine learning, I guess like frameworks, there's this kind of prototypical kind of pipeline that we kind of think through kind of left to right, how we would develop such a thing. So we start completely on the left with our population. So ASD has a prevalence of about 1 in 44 individuals in the general population. Each child through a pregnancy will have a number of different healthcare encounters and various diagnoses, both the mother and the child thereafter. And so all of this data is being collected in numerous ways and numerous sources, and collected in such a way that if we could aggregate that information together and create this really rich profile around an individual, can we potentially develop one of these models? So we need to kind of merge and bring together these large-scale data sets in such a way that we can actually use that as part of the machine learning framework. So that's what we see, this pre-processed data in the middle. And kind of standard to machine learning practice, we would want to segregate that data in a fair way to train the model, to validate it, and then a completely held out data set is used to test it, to determine whether or not the model really performs across a general unseen population. So what we see kind of in the middle, the data formatting is aggregating information from these mom-baby pairs. So that's taking some of the prenatal information from the mother and during the pregnancy, as well as different health encounters and codes and diagnoses, kind of the information we would collect in the neonatal and pediatric period. And you can imagine those look different based on each individual kind of mom-baby pair over time. And really the outcome that we're interested in is whether there was an ASD diagnosis provided at some point in the future, between 18 and 60 months, so 18 months and 5 years, or we would cut off all our data at that 5-year point. And then we do some amount of machine learning. Using that data formatted in that way, we can provide them to these large-scale machine learning models in such a way that we can determine whether the model accurately identifies these individuals. So predicting the increased likelihood of developing ASD. But as most people know, these are black box models. And what's exciting is there's an emergent field in AI known as explainable AI that tries to peer into these boxes, tries to explain how a model focuses in on elements of interest. And so we're considering that as part of this work as well, to ensure that the models that we develop are indeed, identifying features that are important. Next slide, please. And so this is the kind of definition of our study cohort. And so it spans quite a few years and quite a few data sets and a very, very large population. So our, to kind of give you a sense as to how this works, the index date is the infant delivery date. And so our accrual window is between 2006 and 2018. We want to have a minimum 2-year follow-up window of pediatric and neonatal features, but as well as a 2-year look back window for maternal and pregnancy features. And so really the time spans that we're looking at are data that go back as early as April 1st, 2004, all the way up to March 31st, 2021. And of course, as I mentioned, the outcome of interest that we're looking for is whether there was an ASD diagnosis provided after 18 months, but up to 5 years. And really, again, the scale of the richness and diversity of the data we have was quite vast. So we were bringing in data from the Born BIS and NIDA, the CHEO DAD and NACRS data, as well as OHIP and mom-baby data sets. These are just a few amongst several that we had collected together. Next slide, please. And this was basically the scale of what we were looking at. So our final kind of cleaned up version of the entire data set included about 700,000 mother-infant pairs, of which about 10,000 or about 11,000 were indeed ASD cases. So roughly on par with the prevalence we see in the general population. There was a number of exclusion factors that we had to go through to get to this cleanup point, but really this was the bulk of the work. This was creating a framework that was able to link multiple data sets to really organize all the data in such a way that we could use them as part of this framework. No one had really done something of this scale before. So we were excited, at least with the scale of the data that we had, it was, you know, on par with what we would need to train something like a deep learning model. So next slide, please. And so there might be a lot kind of put into the slide. It's not you know, detailed information that we need, but clearly machine learning models require data in a machine learning acceptable format. So we had to encode much of this information. So what we see here on the left, the idea of time series embedding, so for transformer models. A transformer model is kind of the underlying technology we hear of a lot of the new AI methods existent today, ChatGPT and these kinds of things. It's kind of the state-of-the-art architecture for a lot of these studies. And in order to organize our data in such a way that we can pass it to that model and train it, we need to organize all that information kind of on a visit-by-visit basis, numerically encoding a lot of information, organizing that in such a way that we capture kind of the distinct moments between going from maternal features to the neonatal and pediatric features. And in sum, the idea is organizing all of that data in such a way that we can train the model. So here on the right, we cite two different methods. We considered both XGBoost and transformer models. So an XGBoost model is similar to random forest. It's kind of a new contemporary take on the random forest model. But we also wanted to consider the transformer models. And what was unique to our work was we really were experimenting in the architecture, the machine learning architecture behind these methods. And so the idea of creating these large-scale ensembles, so that's taking kind of individually trained component models, having kind of these different experts, we can say, training each of those individually and combining them as kind of like you could think of it as like a panel or a cohort or a vote across multiple different models to come with this you know, high quality or hopefully highly accurate prediction. So that was the model architecture that we had considered. Next slide, please. And what we're excited is with the results. I mean, there was a lot of experimentation over a long period of time to get to this point. It wasn't something we would achieve successfully. So there was a bit of a eureka moment earlier on in the year when we started getting some of these results and playing around with the modeling. So for those who are familiar with these plots, these are receiver operator characteristic curves, are kind of the de facto way of demonstrating performance in machine learning figures. And for those who are more familiar with, say, sensitivity and specificity, we were on the independent test data set, the kind of completely unseen held out data set that representative of the general population. Our model, or the highest ranking model that we had achieved, had a sensitivity of about 70.9% and a specificity of 56.9%. These are all preliminary. This is something that we feel, I mean, in continuing to develop our models, we might be able to even improve upon this further. And so there's kind of an exciting element to that work. Next slide, please. And the goal, really, the long-term goal, I mean, since much of this is kind of pilot work or preliminary studies, is really to refine the long-term vision we would have is, you know, if we were ever to move towards, you know, actually implementing such a model, we would have patients coming in for screening. We would have this well-trained kind of error-minimized ensemble transformer neural network, this thing that we're continuing to refine, with the hopes that it would be able to flag for those specific cases, you know, a higher chance of potentially developing ASDs, so that we could then, you know, intervene or provide resources early. The idea is to make it as early as possible, the early detection and diagnosis, to provide those resources for families and individual support. And the idea around kind of the explainable AI black box models is maybe even being able to understand what is it about, you know, a specific individual that these kind of patient-specific features that helps explain or allows us even to better understand kind of the manifestation of ASD for an individual and across a population. So next slide, please. And so the key takeaways really of this talk, I mean, we're excited to at least report that we have preliminary results that demonstrate the feasibility to identify young children with increased likelihood to develop ASD. We believe our models have face validity. When we start looking at the top ranking features that are most predictive for models, we see certain known ASD risk factors coming out, such as male sex assignment at birth, a low birth weight, some family history factors as well. And really, as I mentioned, we want to continue to refine these models to use kind of explainable AI to better understand and really validate the models through and through. And that's ultimately part of this future work. I mean, to move towards having an AI-based ASD screen, potentially for deployment, and the idea that we could leverage the same framework for similar type studies, that we could look at other types of complex mental health or neurodevelopmental conditions. So next slide, please. None of this would have been possible without this great team. So kind of at the top, myself and Emily were kind of more the computer sciencey AI specialists. We had a whole admin team that was supporting this work, as well as my now director at Bourne, Ontario, Heather Howley, and then a few of the PIs, Stephen Hawken, Mark Walker, and Christine Armour were instrumental to this work. So just want to acknowledge them. And final slide, please. No, I guess that's it. Thank you.
That was it. Yes, thank you so much. Okay, and now we're on to Khaled.
Okay, great. Thank you very much. So the presentation today that I'll give is on generative AI, really with the focus on simulating patients in clinical studies. So in summary, we created a platform to implement these types of simulators, and I'll give you some of the results we've obtained so far. We use data sets that were readily available so we can get results quickly. So a lot of the results I'll show you are from oncology data sets, but they demonstrate the capability, they validated some of our assumptions, and then the next step is to apply those methods for, we're working on applying them to eating disorders data sets, which will be the next phase of the work we're doing. So the next slide, please. So the basic idea behind generative AI is that you train a machine learning model that can then generate new data, new information. And a good example of this is Space Opera Theater, which is an image that was generated or produced by a generative AI model called Midjourney. This particular one won a fine arts competition at the Colorado State Fair about a year ago. And that upset a lot of people because the machine-generated image won against real artists. But just to make the point that the types of images and the types of the quality of generative AI has improved quite dramatically over the last year, to the point that generated images also generate audio, generated video, text, and data has improved quite dramatically. Next slide, please. Another example of generative AI are images of faces. So all of these are fake faces generated by a machine learning model. And various studies have asked members of the public to see if they can tell the difference between real images and fake images. And they couldn't, and they trusted the fake images more than the real images of people's faces. So that's quite interesting. The main point is the quality of the generative images has also improved quite a bit. And it's hard to tell the difference. I mean, if you look carefully at these ones, we took these ones a year ago. If you look carefully, you can tell that something is a little bit off. But overall, they're quite realistic. Next slide, please. So we're using the same type of technology to generate structured data. And the basic idea is that we load in a real data set of some sort, whether it's a clinical trial, a registry, a real-world data set. We train a machine learning model. Different types of machine learning models can be used, some statistical machine learning or deep learning models. And once you train the model, then you can use it to generate new data. And the new data will look very realistic. It'll have the same patterns as the original data. And when you perform analysis, at least in principle, on the generated data, you'll get very similar results as if you were using the real data. One of the advantages of these types of generative models is that you don't need to know what kind of analysis you're going to perform on the data afterwards. So you can create synthetic data sets or machine-generated data sets that are very realistic without knowing a priori how that data will be used. So we've developed a pipeline consisting of our own algorithms and algorithms developed by other people, and we've done a number of evaluations. So next slide, please. One of the basic use cases for synthetic data is data sharing or dealing with privacy issues, because synthetic data sets don't have a one-to-one mapping to real data or to real people. They have strong privacy preserving characteristics. So this slide shows some of the applications of synthetic data, but most of them are around enabling data sharing, data reuse, whether it's internal for research purposes or external for sharing with outside partners, outside collaborators. But if the data is deemed not to be personal information, and regulators are kind of moving in that direction slowly, but surely, considering synthetic data to be non-personal information. So it allows you to share it and make it available to partners with minimal constraints. Next slide, please. So we've done a number of studies where we created synthetic data sets. And one of the questions with synthetic data is whether you get the same results with synthetic data versus real data. So one approach to evaluate that is to take a real data set, create a synthetic version of that, and perform the same analysis on the real and synthetic data sets and compare the results. So that's a good, very practical way of evaluating synthetic data sets. If you're going to share them and make them available to external folks, to partners, or reuse the data because it's more privacy preserving, then you want to make sure that you get the same results. So next slide, please. So one of the studies we did was with 8 breast cancer oncology trials. These were already published. So we managed to get the data, or we had the data sets, and we compared 3 different types of generative models: sequential synthesis, generative adversarial networks, and variational autoencoders. So those are the 3 at the top. And then we replicated the published studies for all 8 of them on the synthetic data sets. And we evaluated three metrics: estimate agreement, decision agreement, confidence interval overlap. So estimate agreement is whether the estimate of the parameters in the synthetic data is within the 95% confidence interval of the estimates from the real data. Decision agreement is whether you have results in the same direction and the same statistical significance on the synthetic as the real data. And the confidence interval overlap is the extent to which synthetic and real data confidence interval for the parameters are overlapping. And in general, we see that sequential synthesis, which is one of the types of generative models, tends to perform quite well with the ability to replicate the published studies accurately across the board with very high confidence interval overlap. Next slide. We also evaluated the privacy risks. So one type of privacy risk is called attribution disclosure. And again, across the board, for all the generative models, the privacy risks were low. So this is, I would say, one of the larger evaluations of synthetic data sets on using real clinical trial data, and the results have been quite encouraging. And we're starting to understand which types of generative models work well and allow you to replicate published results. So it's a good validation of the approach. Next slide, please. The other use case is to simulate clinical trial patients. So the basic problem here, you have low accrual or high attrition in clinical studies, or you can even design studies with fewer patients and then simulate the remaining patients. So we focused on low accrual in the control arm just to start off with. We have other results on both arms, but I'm just going to show you the control arm results. So the basic idea is that we take the same clinical trials and then remove the last X% of patients, and then simulate those back in, and then redo the analysis and compare that to the published results. So go next slide, please. So here what I just said, you take the control arm, you train a generative model from a control arm that didn't recruit enough patients, either deliberately or because of accrual problems, and then simulate those and put them back in, and then compare the augmented clinical trial data sets with the published result that has the real patients. Next slide, please. So here are the results. We looked at 4 different types of generative models: sequential synthesis, GANs, VAEs, and Bayesian networks. And the point 1 and point 2 on the left side show that if we remove the last 10% of the control arm patients and the last 20% of the control arm patients, we kept going all the way down. I'll show you the first sort of the results for the 10 and 20% removal. But let's say the 20% removal, you can see that sequential synthesis and Bayesian networks work quite well. And that on the same metrics we looked at before, estimate agreement, decision agreement, and confidence interval overlap, we're able to replicate the published studies even with up to 20% of the patients missing and simulated back in. We're able to replicate the original studies quite closely. So that's encouraging because it can provide one way to use generative models to simulate patients or augment the data sets with missing patients due to accrual or attrition problems and still get the same results. And this is for 5 of the 8 breast cancer clinical trials that we saw earlier on. Next slide, please. So another study we looked at is mitigating bias. Again, SMA is essentially using synthetic minority augmentation, so using synthetic data to simulate patients. But the basic idea is you have a biased data set where you have a particular group of patients that are underrepresented in a data set. The underrepresentation could be by race, could be by ethnicity, could be by age, could be by sex, or combinations. And under certain conditions, a biased data set will result in biased results. Not always, because if under certain conditions, a biased data set will not give you different results, but in some cases, a biased data set can give you different results. So the idea here is if you know what the correct distribution of this underrepresented group is, then you can train a generative model using the data that you already have and then simulate back the underrepresented group. So we did a simulation with a number of real-world data sets. Where we used the sequential synthesis generative model and evaluated whether this type of bias mitigation would work. Next slide, please. So the basic idea was we do the bias mitigation and compare the results of the analysis to the ground truth. And all the analysis we looked at were logistic regression models. So we were trying to replicate the results of the logistic regression model for the true data set after we do bias mitigation. And we looked at 3 different types of bias: marginal bias, weak conditional bias, and strong conditional bias. So for marginal bias, it would be, for example, certain race is underrepresented. Conditional bias will be on combinations of variables. So for example, certain race with low socioeconomic status. And then we have weak and strong relationships with a conditional relationship. And we compared different types of bias mitigation approach. SMA is the one on the right-hand side. And these graphs, the green part really is what's interesting. It shows to what extent the approach is better, is an improvement over the biased data set. And out of all the methods that are currently looked at or available, SMA, it's not perfect, but it mitigates bias to a good extent. So for low and medium bias, we get very good results. So here we're looking at to what extent the odds ratios for these logistic regression models are an improvement over the biased results for across all the data sets that we looked at under the 3 bias conditions. The next slide, please. And then in this case, we also looked at fairness. There's a number of fairness metrics that evaluate how fair your data is. And again, the synthetic data bias mitigation approach improves the fairness most of the time under different bias conditions, and certainly better than existing approaches that are used today. So we have, again, good results in terms of using synthetic data generation methods for bias mitigation. And so the idea here is we have a platform, we've validated the approach, and now we're going to start applying those, at least initially, to the eating disorders data sets in the next year. Thank you.
Okay, thank you very much, Khaled. This is where the save the date was supposed to be, as you can see. And now I'm going to close down the slides, and we will take questions. And please either raise your hand or put them in chat. I hope you've enjoyed this introduction to the kinds of research that Precision Child and Youth Mental Health sponsored grants have accomplished. So Sarah. You're on mute, Sarah.
Can you hear me?
Now we can.
Sorry, I'm in my car. I'm not driving. So I'm a registered nurse with development and rehabilitation. And my question is for the researcher, and I'm so sorry, I'm terrible with names, doing the AI autism research. Why did you choose 18 months instead of 15 months? Because many children are diagnosed at 15 months. And I also asking this from the perspective of a mom whose son was suspected at 6 months and diagnosed at 15 months. And also a comment on language. We don't develop autism, we recognize autism. We're born with autism. It's not something that we develop over time.
Thank you, Sarah. Kevin.
Yeah, that's me. So thank you for your question. The window that we use, the ASD, I don't know, Christine, if you wanted to, I know you're on the call now, so just to clarify, Christine Armour is the PI of this study, so I'm presenting also on her behalf as well. I don't know if you wanted to take that, or do you want me to comment on it?
Sure. As far as the cutoffs, Sarah, thanks for your comment on language too. That's always helpful to be aware of that. So thank you. And yeah, I believe it has to do with the fact that most of this work was based on a pre-examined cohort of ASD, and so was defined as how reliable that cohort was in terms of diagnosis. So I fully acknowledge your point about timing of diagnosis, and definitely that's something we would look at again in the future, but we had to sort of work within the realms of what we thought would be feasible within a 1-year timeline. But yeah, definitely a good point. Thank you.
And Rob, you have a question. Rob Myatt.
Yes, hi, thank you very much. As for the last speaker, what are the general sizes of these data sets that you need? So the existing sort of the real data to then generate synthetic data?
It depends on the method that's used. The deep learning methods require a lot more data sets to really not overfit and to produce meaningful results. The statistical machine learning methods can work better with smaller data sets, but to give you a more concrete answer, I'd say the minimum would be a few hundred. That's when you start getting good answers, like 200 or so. And of course, if you have thousands of patients, then it works quite well. We have one of the trials had 50 patients, and that's the one that always gives us the most problems. So at that point, we're really hitting the absolute minimum, and it didn't really work very well. The 200 so patient studies were reasonably okay.
And just a follow-up to that is there ever a role for imputation of missing values in a small cohort and then generating synthetic data on top of that? Or would that introduce too many challenges?
The statistical machine learning methods can handle missing values reasonably well, like boosted decision trees, for example. If you use them as the basis for synthesis, they can handle missing data. You don't need to impute. If you're using deep learning, you will have to impute if you want to use all your observations. Will it help or not? You have to be careful because you can't use the same data for imputation as your training. So you kind of have to start cutting up your data in smaller and smaller pieces. So it just depends. The other thing about synthesis is when you synthesize, you want to generate the same missingness patterns in your synthetic data, especially if you want to replicate your original analysis. The original analysis had missing data and was dealt with in a certain way. So your synthetic data should have the same missingness pattern so you can replicate that.
Thank you very much.
Other questions? If not, I have a few I can ask our speakers, but I don't want to hog up the stage here. I'm looking also, you can put it in chat too. Oh, I see. Roger, good morning. Go ahead.
Well, first of all, congratulations, Kathy and the entire team. What a tour de force all of you are doing with this. My question is more for, I guess, Khaled. What is the minimum number you need for some of these smaller represented groups that you're trying to mitigate the risk for? Is there a risk of overfitting? How small is too small that you can replicate when there are, let's say, smaller ends of those underrepresented factors of intersectionality, for example?
Right. So for the bias mitigation use case, you need to have some of the underrepresented patients in the data set so you can train a model. And the idea is once you learn the patterns from these few patients in your data set, then you can simulate more of those types of patients. So it's a similar answer in the sense that you still need ideally a few hundred of patients from the underrepresented group in order to build a good model. So you can do an okay job with smaller numbers, and we're working on getting this to work with smaller and smaller numbers because that's kind of the nature of things. But to get good models, let's say 100 plus at least, maybe a couple of hundred would be the minimal. If you don't have any patients from the underrepresented groups, then you can't do anything. You can use external data sets in that case if you have those available to train a model and use that to do the simulation, but that wasn't the context that we tried. We evaluated the case where you had some of the patients from the underrepresented groups.
Khaled, when you showed us your data where you had worked things out and to see if you could validate it and things, you most frequently, in fact, almost entirely talked about RCTs. I wonder if, many of us do things like case control studies in the very early explorations, trying to see if there's a biomarker that might differentiate between groups. Can the simulation approach work with those kinds of studies, or is it really only best for RCTs?
No, it can work for other types of studies. So the bias mitigation work was on real-world data sets, wasn't on RCTs, but it should work on both. It's just that happened. That was the data set. These were the data sets we had available, and so they were good to test the concepts and the algorithms on. But it doesn't matter. It would work on real data sets as well, real world data as well.
Oh, that's very useful. Thank you. Any more questions? Otherwise, I'll ask another of our presenters. Addo and Christina, sounds very, very interesting and exciting what your future plans are. I just wonder, listening to what you were putting together and trying to put a pilot together, and that you worried about small sample size with your first study that you presented. I wonder if it might be of some use to speak to Khaled to see if there might be a way to help bolster or make your next study perhaps bigger in a way. So
okay, thank you very much. That's a good idea. We'll certainly get in touch with him. Yeah.
Okay, and Kevin, can you explain to us, just so that everybody knows, since most of us, including me, are not familiar with, when you said what needs to happen next with the predictive algorithm that you've developed so far? Do you have to check with clinicians and see if it makes sense? What sort of things happen next?
Yeah, absolutely. I mean, the idea here, and similar to what Christina had said, what was feasible in a year was the idea of kind of bringing together all these data sets in somewhat of a melting pot. We were pulling data set from numerous different sources, but when we think of actually developing a screen, there's a number of kind of layers to how we get to that point, and whether, I think much like Sarah's concern too, I think actually engaging with some of the nursing staff and patient advocates and family advocates is going to be a whole branch of that as well. But from a technical perspective, we have to think what data can we reliably use as part of our screening process, and not all the data that we use. It was a kind of a moonshot idea. Is there even a signal to be picked up on here? So we kind of brought everything together, but would that make a reliable screen? If we can't rely on having all those kind of data points available all the time for every single person, we can't deploy a screen. So I think there's going to be a lot of work with clinicians working through what exact data points, we call them in the machine learning community different features, like the different kind of variables that we want to, or covariates that we want to actually include, that we can reliably include, and then actually evaluate, bringing that closer and closer to an actual trial to evaluate in an existing population is going to be the long-term process. So there's a lot of kind of technical kind of community engagement. There's a lot of clinical consultation, I think, that we would need as well, just to get to a point where we're incrementally moving towards what would be a reliable screen. It's the long-term idea, but at least which was why this was a lot of kind of preliminary work, just to see if there was something to be detected. So
yeah. So it sounds like there's some work on the constructs, and there's also some practical work, which Sarah certainly pointed us in the direction of too.
Yeah, absolutely.
Well, if no one else has questions, what I just want to say for people who are on, just as a way of explanation, you've heard all of our investigators talk about only having a year. And in fact, we did only have 18 months to use $5 million, which you might think might be a dream, but if it's also very difficult. So these people and all Mark Norris's group and Mark Tremblay's group and the other folks have worked incredibly hard to get results in 18 to 20 months. So, and I just want to call out all their hard work. So we'll close down for now because it'll give everybody time to move to their next meeting. And we've really enjoyed sharing what we're doing with you. And we'll try to make sure that we keep everybody posted and lots of things that are coming down the line. So thanks very much, everybody, and have a good day.